Hello, everyone. I'm Yaara Erez from the Faculty of Engineering and Gonda Multidisciplinary Brain Research Center at Bar-Ilan University in Israel. Today, I'm going to talk about our efforts toward individualized cross-modality imaging of executive control in clinical research. Much of this work was done at the University of Cambridge while I was still based there, and I have no disclosures to report. Executive control is a collection or family of cognitive processes, including attention, planning, problem solving, task switching, information processing, and so on, that we use all the time in our everyday life. Just as an example, in this kitchen service, the people in the picture need to employ all of these cognitive processes, even if their vision works perfectly well, and if their hands work perfectly well. If their executive control functions don't work well, they can't perform this kitchen service. And the same applies to everything that we do in our daily life, packing a bag, getting to this conference in the right day and time, planning a holiday or a family dinner. At the neural level, executive control is supported by a frontoparietal network, also termed the multiple demand network, to reflect its domain general nature. You can see this network here, which has been recently updated with the HCP data set, showing it in a finer resolution. This is a bilateral network that is recruited whenever cognitive demand increases, and it's a very robust and consistent evidence across many neuroimaging studies. This network has been associated with a wide range of executive control processes. In the talk, I'm going to talk about two main projects or studies. I will start with fMRI data from healthy participants, where we look at individual differences in how this network is localized for the different participants. And I will continue to study in patients and our efforts to map executive control regions with ECOG and further with fMRI data in patients with brain tumors. I will start with fMRI data in healthy participants and how we can localize the frontoparietal network in individuals. The study was led by my former students, Sneha and Flurja. And to identify this network in individuals and also say something about the signal that we can get from the individually defined regions, we used three tasks commonly used in the literature: a spatial working memory task, a verbal working memory task, and a variation of the Stroop task. I'm showing here the patterns of activity at the group level, and we can see this typical activity of the frontoparietal network, which is a good sanity check. But what we were really interested in is what happens at the individual level. These are examples from 6 different participants, and you can see this typical pattern of activity in all 6 participants, but you can also see differences between them. I highlighted just a couple of places, but there are more. So we set to quantify these differences. What I'm showing here is a probability map on the left, where the color of each voxel indicates the number of participants that had a significant change in activity with increased demand. For each of the 3 tasks, we had 2 conditions, an easy and a hard one, and we look at the change in activity between them. And we can see these yellower or brighter hotspots with a typical pattern of activity of the frontoparietal network. But we can also appreciate how widespread this activity is across participants for the more reddish and darker voxels. We then looked at the reliability of activity for each individual participant using a voxel-wise correlation. Each of the participants repeated each of the tasks twice, and we looked at the correlation across the 2 runs. And for each of the tasks, we can see quite high reliabilities up to 0.75, almost 0.8, which is quite remarkable for fMRI data. And when we repeated the same analysis, this time correlating activity across voxels between different participants, we can see this huge drop in correlation reflecting the variability across individuals. So we can identify this network in individuals quite reliably. What we ask next is whether this reflects something more general about executive control. Following work by Ev Fedorenko, we used anatomically constrained maps to individually localize executive control regions in individuals using each of the three tasks as a functional localizer. And we used a fourth criterion task, a task switching task, another core aspect of executive control, to look at activity within these individually defined regions. So as a start, we used the group template to define the network in the same way for all individuals. What we expected to see is an increase in activity in switch compared to state trials, and we were able to replicate previous findings. But when we looked at the individually defined regions, not only we could see this same effect, but it was much stronger. Meaning that the individually localized regions are more generally related to executive control, and furthermore, they demonstrate a larger sensitivity. And we repeated the same approach in several studies, including MVPA study with an attention task, another core aspect of executive control, and we're able to show that only relevant information, but not irrelevant one, was represented across this network in the individually defined regions. So we can reliably localize these regions in individuals, and this is very important so we can make sure that the signal that we look at is indeed from the right places in each individual. And this has some implications for clinical research, because when we work with patients, what we really want is to offer the best treatment and diagnosis for the patient that sits in front of us, and not to the average non-existing patient. And this leads me to the second part of the talk, when I will show you some data from ECOG and fMRI in patients with brain tumors. We worked in this project with patients with diffuse gliomas. These are tumors that are slow growing and non-aggressive, but they infiltrate into the nearby tissue. So during surgery for the removal of the tumor, neurosurgeons need to optimize a fine balance between removing as much as possible of the tumor and preserving function. Any function, and this is usually done primarily for motor function and some aspect of language using a functional mapping procedure in an awake surgery. So the patient is awakened after the skull has been opened during their surgery, and then they are asked to perform different cognitive tasks, and the neurosurgeon applies brief pulses of stimulation, electrical stimulation on different parts of the brain. The idea is that if stimulation leads to impairment, reversible impairment, for example, the patient stops talking, then the stimulated area may be critical for this function, in this case, speech production, and the neurosurgeon might decide not to resect it, even if from an oncological point of view, it would have been better to take this part out. So for executive function, this is slightly more complicated, and so far there are only a handful of studies using this approach in the literature. And this is primarily for two reasons. First, the complex nature of executive control, and second, the distributed network that supports them. So our approach was to use ECOG, electrocorticography, during awake surgery to identify a functional signature of executive control regions, so we can guide neurosurgeons to use stimulation in the right places, and and make this functional procedure mapping more efficient and safer. We place the electrodes during the surgery when the patient is awake and performs cognitive testing. And we ask them to do two tasks, counting tasks, simple counting 1 to 20, and a harder one, so increased cognitive demand counting while switching between letters and numbers, 1A, 2B, 3C, with the aim of recruiting executive control regions similarly to what we see in fMRI studies, and identify this functional signature. And this study was done as part of a large collaboration and mainly with my former student, Muatas. What we looked at first was activity in the high gamma range, which has been previously shown to correlate with the fMRI bold response and neural processing more generally in other domains. And we were able to see increases in high gamma with increased cognitive demand, so difference between the hard and the easier task. This is the red blob that you see in the frontal cortex, where we expect to see the recruitment of executive control regions. What I'm showing here is activity in individual electrodes. This is data across all participants, first to establish this idea that we can actually use this data or this measure to identify executive control regions. We also looked at activity in the beta range, where we expected to see decreases in the beta range with increased demand based on previous literature. And what we noticed is that the high gamma changes were quite focal, while the beta changes were quite widespread. So we set out to quantify these changes in high gamma activity with increased demand in more detail. And for each electrode, we determined whether the network that these electrodes belong to based on its location and the HCP parcellation. And then we used several measures to look at activity in electrodes in the different networks. I'm showing just a few. So here I'm showing the percentage of electrodes with significant changes in activity. And we can see in the yellow bar that increases in high gamma, which are much more likely within the frontoparietal network compared to non-frontoparietal electrodes in the gray bar, and in the beta range, decreases were equally likely both within and outside the network. For the percentage of overall power change across all electrodes, we see a similar pattern of results, increases in the within the frontoparietal network in the high gamma, decreases both within and outside the network in the beta range. We then zoomed in on these changes in high gamma activity, and we looked at each of the frontal networks separately. And what we found is that this high gamma activity was specific only to the frontoparietal network, and we didn't see it in any other network. So high gamma activity distinguishes frontal control regions from adjacent networks. And we think that these results show that we can use high gamma activity as a functional signature to identify executive control regions. And what we are looking at now is whether we can do that reliably in individual patients. What I showed you so far is data from healthy tissue. So electrodes were placed further away from the tumor in what is considered to be a healthy appearing tissue. But we have evidence from the last few years that show tumor-neuron interactions and how they remodel neuronal circuits at multiple scales. This is just one example from a study showing changes between normal appearing and glioma-infiltrated cortex with respect to speech processing. What Ayan was interested in is how activity in tumour-infiltrated cortex based on these previous studies is implicated and how this is related to whole brain large-scale connectivity with networks as can be measured with resting state fMRI from before the surgery. So we used both the ECOG data during the surgery and resting state data from before the surgery. I'm showing here examples from 2 patients with the data from the surgery, electrodes that were showing significant change in activity or a signature of executive control activity, and how they correspond to canonical functional connectivity with canonical networks. We have some data also linking it to cognitive outcome. And if you want to hear more, I will be presenting Ayan's work in a poster in this conference, and you are most welcome to come and have a discussion. To sum up, I showed you that the executive control network is unique in individuals and can be reliably localized. And I then showed you some work in patients with brain tumors looking at neural signature of executive control with ECOG and how this may be related to whole brain functional networks and connectivity as can be measured with resting state fMRI. And we are now continuing this line of work, translating what we know and the methods that we use in basic science to better understanding of how functional networks are organized and change over time in patients with brain tumors. Thank you very much for your attention. Thank you for all of my collaborators and students, and of course, the patients.